All right, so your homework tonight is to watch this video where I talk about this process called blue-white screening, which is a method in which we determine if a gene has been inserted into a plasmid or not. Now in class we talked a little bit about plasmids, so let me just review again what plasmids are. They're small extra circular pieces of DNA that bacteria have. They're not their main circular piece of DNA, it's an extra one. And we're actually going to use these things because they can be taken up by bacteria. They're like that, remember I mentioned that bacteria can transform by picking up things from genes from their environment. This is how they do it, is through a cloning vector called a plasmid. But what we're going to try to do is we're going to actually clone a gene in these plasmids. We're going to copy a gene, we're going to put it in there, and then the bacteria is going to make copies of that specific gene when it copies itself, and also make whatever the gene product of that protein is. For example, like the insulin gene. Remember, the way this works is you will actually take restriction enzymes and cut out the gene that we want to be inserted. Now, in prokaryotes, you can cut it directly out of the DNA. So if there's a prokaryotic gene we want to transfer from one prokaryote to another into this plasmid, then we can do so fairly easily by cutting it out using a restriction enzyme that flanks the gene. But in eukaryotes, there's complicated parts to it, right? Because the DNA has lots of introns and exons that bacteria cannot remove or prokaryotes cannot remove. So what we have to do is we have to turn it into mRNA first, and then we use reverse transcriptase to turn it back into DNA, and then we will cut it with restriction enzymes and then put it into our bacteria. Okay. We will then add some ligase, which will help to stick it, put it in. Remember, it should just, because it has the same sticky ends. If you cut with the restriction enzyme both the DNA that we want inserted and the plasmid itself, the sticky ends should match up. So it should stick or anneal. And then we use ligase to solidify the backbone again to kind of hold it in place properly. And then we've created our recombinant DNA, or our plasmid, which is what recombinant DNA is essentially the plasmid with our new gene that has been inserted into it. And here's showing you that here, kind of in a simplified version, with the gray part being the gene that we're inserting, and then the green part being a subsection of the plasmid from the bacteria. Okay, so now that you know the basics of it, right, and reviewed the basics, I talked about how DNA can be picked up by bacteria from their environment. That process is called bacterial transformation. That's how bacteria can pick up resistance to antibiotics, for example, is through that process. That is how, remember, that virulent strain in the Griffith experiment picked, or the non-virulent or avirulent strain picked up the virulence from the dead bacteria, is that while it spilled its plasmids out, it gave it its dangerousness, and it picked those up, and then it became dangerous. So the way this is going to work is we're going to have some special genes inside of our plasmid. Now remember, we make these plasmids in the lab. Okay, these plasmids are special plasmids we have genetically engineered or designed. Okay, so we are actually going to do a selective process, which I'm going to kind of explain, and you're going to have to follow me because it's kind of hard, and then we're going to do an activity in next class to explain how this whole thing works. All right, so let's talk a little bit about blue-white screening, which is the process in which we take a plasmid that has some selective genes on it that determine whether or not, A, if the plasmid has been picked up, which is designated by this ampicillin or this kanamycin bacterial resistance, antibiotic resistance gene. That means that if they have that gene, they're resistant to that specific antibiotic. And the LAC-Z gene, which is basically the gene that codes for the enzyme beta-galactosidase, which is an enzyme that breaks down lactose, but also breaks down a special sugar called XGAL, which we're going to talk about in a second. Okay, so what we are going to do is we are going to, when we insert our gene, when we cut it, we're going to put it into the LAC-Z gene, or the beta-galactosidase enzyme gene. And this specialized gene actually has restriction sites for almost any restriction enzyme you would want. When it is intact and a gene has not been inserted, we will actually code for this enzyme. We'll code for beta-galactosidase. That's going to be an important thing. If we do not have that enzyme, we will not code for beta-galactosidase. So beta-galactosidase will be disrupted. If our gene is inserted, beta-galactosidase is disrupted. That's going to be important. Notice the ampicillin has nothing to do with this, the resistance gene, just this. Okay? So then, how does this work? Remember, I said the LAC-Z gene codes for an enzyme called beta-galactosidase. That is, again, part of the lac operon, and that's involved in breaking down lactose. It's another name for lactase, the enzyme. It also breaks down a special sugar called XGAL, and XGAL actually, when it's broken down, turns blue. So when beta-galactosidase is present, the bacteria is going to turn a blue color. Okay? When beta-galactosidase is not present, it's not going to break down XGAL, and it's going to stay white. Hence the blue-white screening part. Okay? So if restriction enzymes disrupt the LAC-Z gene, because the clone gene is inserted into the LAC-Z gene, you will not get beta-galactosidase because you're disrupting the gene, and you're not going to get XGAL, and you're not going to get blue color. It's going to stay white. But if you have not inserted it, if it's not inserted properly, it will be blue. So we want to select for the bacteria that is actually white. The white bacteria is going to be the one that has our gene inserted properly. It has picked up the plasmid and inserted the gene properly. So how do we know that the bacteria didn't just pick up the plasmid, right? So plasmidless bacteria can also not break down LAC-Z. Also, that other gene, that ampicillin resistance gene, was used to determine whether or not it picked up the enzyme at all, or excuse me, picked up the plasmid at all. So if in our agar plates, which are the plates where the bacteria sit with nutrients in them, we add XGAL sugar, which will turn blue if it's cleaved, and ampicillin, which is going to select for if the bacteria picked up the plasmid or not, only bacteria with plasmids will grow. And only bacteria that have the gene you want to clone, right, whatever it is, let's say it's the insulin gene, right, if that's been, it'll disrupt that beta-galactosidase gene, and it will be a white. It will stay white because it disrupted it. If it stayed whole and it wasn't inserted properly, it would be blue. So essentially, we would look at this plate. Notice we put bacteria over this whole thing. Anywhere there's not either blue or white was bacteria that died because it didn't pick up the plasmid. Each of these all picked up plasmids. The difference is that the white ones picked up ones with the gene inserted that we wanted, and the blue ones did not. So that's how we know whether or not a gene has been properly inserted into our plasmid that we're trying to clone, and whether or not the plasmid has been taken up by the bacteria. If you have any questions, we're going to go over them tomorrow in class, and we're going to do an activity where we kind of simulate this process.